Now, let's discuss about more modern systems biology. Systems biology means the study of interactions among the components of biological systems and how these gives rise to the function and behavior of the whole system. So systems biology requires more of a quantitative, mathematical, or computational approach. And this is more holistic approach than previous, maybe decades ago, reductionist biology approach. Reductionist approach means you take out the part and part, we study this part well enough, then you may be able to construct the whole picture. But systems biology is thanks to the development of more technology. Instead of like single gene study, we are now dealing with a whole genome study. And this big data, we can actually build a network or modeling of how the whole system components are interacting together. So this is a diagram for systems biology. So in life science, starts usually with hypothesis. So in many life science grant, you have to start with an hypothesis. What is hypothesis? Hypothesis is a worldview that is testable. So that with your hypothesis, you test your hypothesis either that way or the other way. And for example, you are curious about one gene in a biological system. You consider this gene may be important for some function, then you could either eliminate that gene and see how the system, the cell, or the animal works. Or if the gene, you can actually put the gene more here so that see if that also affects the final expression and function. And you can have your hypothesis. And by this genetic modification, and you measure the outcome with quantitative measure, and life science can study one gene at a time. However, now in this genome area, we can actually study multiple genes simultaneously. So that requires a systems science approach. So we see the biological entity as a system that requires a modeling and synthesis and analysis of the whole system. So which led to information science component. So now we are building a lot of databases, and to study those databases, we need to build a model. So we need a modeling tool, and instead of one, two, or few, we are dealing with hundreds and thousands of all this data. So we need to come up with a better way how to visualize the results. So this is possible by advanced array technology. So array is just a grid pattern, which the measurement, instead of one single measurement, we are talking about simultaneous hundreds and thousands of measurements. So simultaneous measurements of thousands of genes or proteins within a cell or tissue. And this can be studied by the advance of bioinformatics. So bioinformatics is the application of statistics and computer science to the field of this molecular biology. And that has been associated many disciplines. So I want to give you an example that, so DNA sequence, a single DNA study, a gene study, we call genetics. Now we are talking about the whole genes, so that allows us to study the whole genome. So it's called genomics. And protein expression and the whole number of protein study, we call proteomics. And gene expression, usually from the DNA into the RNA expression, we call gene expression. And that DNA to RNA conversion is called transcription. So this gene expression of study is called transcriptomics. This omics means the whole gene expression. And if we are interested in metabolites, the whole metabolite study is called metabolomics. If we are interested in carbohydrates, then the study of the whole carbohydrates as a glycomics. If we are interested in lipids, and we call it lipidomics. And to deal with this gene expression, I just bring one example here. So each dot means the gene expression of one part. So this green and red is called a heat map. So that compared to the relative average, if the expression is higher, you give more reddish color. It's lower than average, you give, in this case, it's a green color. And the algorithm makes all of this to be arranged so that you can see similar parts to be similarly arranged. So these gene expression values are from microarray experiments, and one can use heat maps to visualize the results of data analysis. This allows us to comprehensively understand what's going on in a not single level, but the whole number of thousands of experiments. So let's summarize. So this chapter of what is biomedical engineering, we covered the role of biomedical engineers for the development of reliable, safe, and inexpensive medical technology to improve the life expectancy and the quality of our lives. So biomedical engineering or medical engineering is the application of science, mathematics, and engineering design principles ultimately to improve our human health. And this biomedical engineering often specializes in various sub-disciplines or fields. So whatever your backgrounds are, there are always you can contribute as a biomedical engineers to solve ultimate problem. So emerging human diseases and our new discoveries in physiology and human health provides us to new problems of the future. So with the advancement of engineering and science and sub-disciplines, we can combine to cope with challenging problems of human health. Thank you for your attention.